Chapter 1: Foundations of Animal Behavior Study. Animal behavior, known scientifically as ethology, represents one of the most captivating and complex fields of biological study, encompassing the systematic investigation of how animals interact with their environment, communicate with one another, find food, avoid predators, reproduce, and navigate the countless challenges of survival. This discipline bridges multiple scientific domains including neuroscience, ecology, evolution, psychology, and genetics to understand the mechanisms underlying behavioral patterns that have evolved over millions of years. The study of animal behavior provides crucial insights not only into the lives of other species, but also into the evolutionary origins of human behavior and the fundamental principles that govern all living systems. The modern science of animal behavior emerged from the convergence of several intellectual traditions, most notably the work of European ethologists like Konrad Lorenz, Nicolaas Tinbergen, and Carl von Frisch, who pioneered observational studies of animals in their natural environments. These researchers rejected the laboratory-confined approaches that had dominated early 20th century psychology, instead emphasizing the importance of studying behavior in ecological contexts where natural selection had shaped behavioral adaptations. Their revolutionary approach recognized that animal behavior could only be properly understood when observed under the conditions in which it evolved. Lorenz's groundbreaking work on imprinting demonstrated how young animals form crucial social bonds during critical periods of development, revealing that learning and innate programming work together to produce adaptive behaviors. His observations of greylag geese, following the first moving object they encountered after hatching, illustrated the precise timing mechanisms that ensure offspring form appropriate attachments to their parents. This discovery fundamentally changed scientific understanding of the relationship between genes and environment in behavioral development. Tinbergen's 4 questions provided a comprehensive framework for understanding any behavioral phenomenon by examining its immediate causation, developmental history, evolutionary function, and phylogenetic origins. This approach, known as Tinbergen's 4 whys, revolutionized the field by demonstrating that complete understanding of behavior requires analysis at multiple levels, from the neural mechanisms that control immediate responses to the evolutionary pressures that shaped behaviors over geological timescales. The distinction between proximate and ultimate causation became central to behavioral analysis, with proximate causes addressing the immediate mechanisms and developmental processes that produce behavior, while ultimate causes examine the evolutionary functions and historical origins of behavioral patterns. This framework allows researchers to investigate both how behaviors work at the mechanistic level and why they exist from an evolutionary perspective. Classical ethology emphasized the importance of fixed action patterns, stereotyped behavioral sequences that are triggered by specific environmental stimuli called sign stimuli or releases. These innate behavioral programs demonstrated that much animal behavior follows predictable patterns that have been refined by natural selection to produce adaptive responses to recurring environmental challenges. The study of fixed action patterns revealed the sophisticated programming underlying apparently simple behaviors. The concept of critical periods in behavioral development highlighted how early experiences during sensitive phases of an animal's life can have permanent effects on later behavior. These developmental windows represent times when the nervous system is particularly plastic and responsive to environmental input, allowing animals to fine-tune their behavioral repertoires to local conditions while maintaining the basic structure provided by genetic programming. Behaviorism, which dominated American psychology during much of the 20th century, initially provided a competing paradigm that emphasized learning and conditioning, while largely ignoring evolutionary considerations. However, the eventual synthesis of ethological and behaviorist approaches led to a more complete understanding that recognized both the importance of learning mechanisms and their evolutionary foundations. This integration demonstrated that learning itself is an evolved capacity that has been shaped by natural selection. The field of sociobiology, pioneered by E.O. Wilson, brought evolutionary thinking to bear on social behaviors, demonstrating how apparently altruistic behaviors could be understood in terms of genetic relationships and inclusive fitness. This approach sparked considerable controversy, but ultimately provided crucial insights into the evolution of cooperation, competition, and social organization across diverse animal taxa. Modern animal behavior research employs sophisticated technological tools, including GPS tracking, molecular genetic analysis, neurophysiological recording, and computational modeling to investigate behavioral phenomena with unprecedented precision and scope. These advances have revealed previously hidden aspects of animal cognition, communication, and social organization, while opening new frontiers for understanding the mechanistic basis of complex behaviors. The integration of animal behavior with conservation biology has become increasingly important, as human activities continue to alter natural environments at unprecedented rates. Understanding behavioral ecology is crucial for predicting how species will respond to habitat fragmentation, climate change, and other anthropogenic challenges, while developing effective conservation strategies that account for behavioral needs and constraints. Animal welfare science has emerged as another important application of behavioral research, using knowledge of natural behavior patterns to improve conditions for animals in captivity, agriculture, and research settings. Understanding species-specific behavioral needs provides the foundation for creating environments that allow animals to express natural behaviors while minimizing stress and suffering. The comparative approach in animal behavior research examines similarities and differences across species to understand both universal principles and unique adaptations. This method has revealed fundamental commonalities in behavioral mechanisms, while highlighting the remarkable diversity of solutions that different species have evolved for similar problems. Comparative studies provide crucial tests of evolutionary hypotheses about behavioral evolution. Contemporary animal behavior research increasingly recognizes the importance of individual differences in behavior, moving beyond species-typical descriptions to examine behavioral variation within populations. This approach has revealed the existence of animal personalities, consistent individual differences in behavior that persist across time and contexts, providing new insights into the evolution and maintenance of behavioral diversity. The field continues to evolve rapidly, as new technologies and theoretical frameworks provide fresh perspectives on classical questions, while revealing entirely new phenomena. Current research frontiers include the study of animal cognition and consciousness, the neural basis of complex social behaviors, the role of epigenetic mechanisms in behavioral development, and the behavioral responses to rapid environmental change in the Anthropocene. Chapter 2: Communication and Social Behavior. Communication forms the foundation of animal social life, enabling individuals to coordinate activities, share information about resources and dangers, establish relationships, and engage in the complex interactions that characterize group living. Animal communication systems range from simple chemical signals that convey basic information about identity and reproductive status, to sophisticated vocal languages that can transmit detailed information about events occurring at distant locations. Understanding these communication systems reveals the remarkable diversity of ways that animals have evolved to share information, while highlighting the cognitive abilities that underlie complex social interactions. Chemical communication represents the most ancient and widespread form of animal signaling, utilized by organisms from bacteria to mammals to convey information about identity, reproductive status, territorial boundaries, and environmental conditions. Pheromones, chemical signals that trigger specific behavioral or physiological responses in conspecifics, provide a reliable means of communication that can persist in the environment long after the signaler has departed. These chemical messages can be incredibly specific, conveying not only species identity, but also individual recognition, genetic compatibility, and social status. Many mammals use complex scent marking behaviors to establish territorial boundaries and communicate social information to other individuals in their area. Wolves, for example, use urine marking to indicate pack territory, while also conveying information about pack size, reproductive status, and recent activities. These chemical signals provide a form of time-delayed communication that allows animals to gather information about their social environment even when other individuals are not directly present. Acoustic communication has evolved independently in numerous animal lineages, producing diverse sound-based signaling systems that can transmit information rapidly over considerable distances. Birdsong represents perhaps the most studied example of acoustic communication, with many species producing complex vocalizations that serve multiple functions including territory defense, mate attraction, and species recognition. The development of birdsong involves both genetic predispositions and learning processes, with young birds typically learning song patterns from adult tutors during critical periods of development. The discovery that some bird species can learn and modify their vocalizations throughout their lives revealed unexpected parallels with human language development, while demonstrating the sophisticated cognitive abilities underlying apparently simple behavioral patterns. Songbirds show regional dialects, cultural transmission of vocal patterns, and the ability to create new song variants through improvisation and recombination of existing elements. Cetacean communication has provided some of the most compelling examples of complex acoustic signaling in non-human animals. Dolphins develop signature whistles that function as individual names, allowing for recognition and calling of specific individuals within social groups. Humpback whales produce elaborate songs that can last for hours and travel across ocean basins, with song patterns showing cultural evolution as modifications spread through populations over time. Visual communication encompasses a vast array of signals including body postures, facial expressions, color changes, and elaborate displays that convey information about aggressive intentions, mating readiness, and social status. Many animals have evolved specialized visual signals that are precisely tuned to the sensory capabilities and ecological needs of their intended receivers. The bright coloration of many tropical fish, for example, serves as a form of rapid visual communication in environments where chemical and acoustic signals might be less effective. The peacock's tail represents one of the most famous examples of visual sexual signaling, with males producing elaborate displays that demonstrate their quality as potential mates. These displays involve not only the spectacular tail feathers, but also coordinated movements and positioning that maximize the visual impact on observing females. The evolution of such elaborate signals demonstrates how sexual selection can drive the development of communication systems that may seem maladaptive from a survival perspective, but provide crucial advantages in reproductive competition. Many animals combine multiple sensory modalities in their communication systems, producing multimodal signals that provide redundant information, while also allowing for communication across different environmental conditions. Honeybee waggle dances combine tactile, acoustic, and chemical elements to convey detailed information about the location and quality of food sources, enabling colony members to make informed decisions about foraging activities. Deceptive communication represents a fascinating aspect of animal signaling that reveals the complex strategic thinking underlying many behavioral interactions. Some animals have evolved to produce signals that mimic those of other species, either to avoid predation or to gain access to resources. Bolus spiders, for example, produce chemical signals that mimic the sex pheromones of moth species, attracting male moths that become prey rather than finding mates. The evolution of communication systems involves complex trade-offs between the benefits of effective signal transmission and the costs of signal production, and the risks of eavesdropping by unintended receivers. Many animals have evolved private communication channels that are difficult for predators or competitors to detect, such as ultrasonic vocalizations or electrical signals that operate outside the sensory range of most other species. Social organization in animal groups emerges from the complex interactions between individual behavioral strategies and the constraints imposed by ecological conditions. Group living provides benefits including enhanced predator detection, cooperative foraging, and shared care of offspring, but also creates costs including increased competition for resources and greater disease transmission. The balance between these costs and benefits determines the optimal group size and structure for different species in different environments. Dominance hierarchies represent one of the most common forms of social organization, with individuals establishing rank relationships that determine access to resources and mating opportunities. These hierarchical systems can reduce the frequency of aggressive conflicts by establishing clear expectations about which individuals will defer to others in competitive situations. The maintenance of dominance relationships often involves complex signaling systems that allow individuals to assess relative fighting ability without engaging in costly physical confrontations. Cooperative breeding systems have evolved in numerous animal species, with some individuals helping to raise offspring that are not their own. These systems initially presented evolutionary puzzles, since helping behavior appears to reduce individual reproductive success. However, research on the genetic relationships between helpers and offspring has revealed that such systems often involve close relatives, allowing helpers to increase their inclusive fitness by assisting in the reproduction of individuals who share their genes. The study of animal societies has revealed remarkable parallels with human social organization, including division of labor, cultural transmission of behaviors, and sophisticated decision-making processes that involve integration of information from multiple individuals. Honeybee colonies, for example, make collective decisions about nest sites by combining information gathered by scouts and using democratic processes that typically result in selection of the highest quality option. Understanding animal communication and social behavior has important implications for conservation efforts, as many species depend on complex social interactions for successful reproduction and survival. Habitat fragmentation can disrupt communication networks and social structures, leading to population declines, even when adequate resources remain available. Conservation strategies increasingly recognize the importance of maintaining social connectivity and communication networks in animal populations. Chapter 3: Foraging and Feeding Behaviors. Foraging behavior encompasses the complex decision-making processes and behavioral strategies that animals employ to locate, capture, and consume food resources while minimizing energy expenditure and reducing exposure to predation risk. The study of foraging behavior has revealed that animals act as sophisticated decision-makers who must constantly evaluate trade-offs between energy gain, time investment, safety considerations, and opportunity costs in dynamic environments where resource availability and predation pressure vary unpredictably. Understanding these behaviors provides crucial insights into the ecological relationships that structure natural communities, while revealing the cognitive abilities that enable animals to survive in challenging and competitive environments. Optimal foraging theory provides a quantitative framework for understanding how natural selection shapes feeding behaviors, by predicting that animals should forage in ways that maximize their net energy intake, while minimizing costs and risks. This approach treats foraging as an optimization problem where animals must make decisions about which prey items to pursue, how long to remain in particular feeding patches, and when to move between different foraging areas. While real animals rarely achieve perfect optimization, the predictions of optimal foraging theory often provide good approximations of observed behaviors. The marginal value theorem represents one of the most successful applications of optimal foraging theory, predicting how long animals should remain in resource patches before moving to new areas. According to this model, foragers should leave a patch when the instantaneous rate of energy gain falls below the average rate that could be obtained by traveling to and exploiting a new patch. This prediction has been confirmed in numerous species, from bumblebees visiting flowers to birds feeding on insects in different trees. Diet selection models examine which food items animals should include in their diets based on the energy content, handling time, and encounter rate of different prey types. These models predict that animals should specialize on high-quality prey when such items are abundant, but should broaden their diets to include lower-quality items when preferred prey becomes scarce. The predictions of diet selection models have been supported by studies of predators ranging from shorebirds selecting among different shellfish species to primates choosing among various fruit types. Central place foraging occurs when animals must return to a fixed location such as a nest, den or territory center after each foraging trip. This constraint creates additional optimization challenges, since foragers must balance the benefits of traveling farther to reach higher quality resources against the increased travel costs and reduced time available for actual feeding. Central place foragers often show distance-related changes in their selectivity, becoming more choosy about which items to collect as they travel farther from their central place. Group foraging strategies have evolved in many species as a means of improving foraging efficiency through information sharing, cooperative hunting, or reduced predation risk. Flocking birds and schooling fish can benefit from the collective vigilance of group members, while also taking advantage of information about food discoveries made by others. However, group foraging also creates costs, including increased competition for food items, and the potential for individuals to exploit the efforts of others without contributing to group success. Information use in foraging involves complex cognitive processes through which animals gather, store, and utilize knowledge about resource distribution and quality. Many animals maintain detailed spatial memories of productive foraging locations, and can adjust their behavior based on past experience and current environmental cues. Honeybees, for example, use landmark navigation and internal compasses to return to profitable flower patches, while also sharing information about resource locations through their sophisticated dance communication system. Territorial behavior represents one strategy for securing reliable access to food resources by defending areas that contain predictable supplies of high-quality food. Territory holders must invest energy in defending their areas against intruders, but successful territorial defense can provide exclusive or preferential access to contained resources. The economics of territorial defense depend on resource distribution, competitor density, and the costs of aggressive interactions. Cache hoarding behavior has evolved in numerous species as a strategy for dealing with temporal variation in food availability. Animals that cache food items must make complex decisions about which items to store, where to place caches, and how to retrieve stored items while minimizing losses to pilferage by other animals. Cache recovery often requires sophisticated spatial memory abilities that allow animals to relocate dozens or even hundreds of hidden food stores. Predator-prey relationships create complex evolutionary arms races that shape both hunting strategies and anti-predator defenses. Predators have evolved diverse hunting techniques including active pursuit, ambush tactics, cooperative hunting, and tool use that increase their success in capturing prey. Simultaneously, Prey species have evolved defenses including cryptic coloration, warning signals, group vigilance, and escape behaviors that reduce their vulnerability to predation. The coevolution of predators and prey has produced remarkable examples of behavioral adaptation, including the high-speed chases between cheetahs and gazelles, the sophisticated echolocation systems of bats and the acoustic defenses of their moth prey, and the complex chemical warfare between plants and herbivorous insects. These evolutionary interactions continue to drive innovation in both offensive and defensive strategies. Tool use in foraging has been documented in an increasingly diverse array of animal species, from the stick tools used by chimpanzees to extract termites from mounds to the rocks used by sea otters to crack open shellfish. These behaviors demonstrate sophisticated problem-solving abilities and often involve cultural transmission of techniques between individuals. The study of animal tool use has provided important insights into the cognitive prerequisites for technology and the evolutionary origins of human technological abilities. Seasonal and migratory foraging strategies allow animals to track resource availability across time and space, often involving remarkable feats of navigation and timing. Many animals migrate vast distances to take advantage of seasonal peaks in food availability, while others employ seasonal changes in diet or foraging behavior to cope with predictable fluctuations in resource availability. Arctic terns migrate from Arctic to Antarctic regions annually, following peak productivity seasons in both hemispheres. Human impacts on animal foraging behavior have become increasingly significant as habitat modification, pollution, and climate change alter resource availability and distribution. Many animals have shown remarkable behavioral flexibility in adapting to anthropogenic environments, with some species learning to exploit human-provided food sources, while others suffer from disruption of traditional foraging patterns. Understanding these impacts is crucial for developing effective conservation strategies. The study of foraging behavior continues to evolve as new technologies allow researchers to track individual animals with unprecedented precision and detail. GPS tracking, accelerometry, and biologging devices now provide continuous data on animal movements and behaviors, revealing previously hidden aspects of foraging strategies and decision-making processes. These technological advances are revolutionizing understanding of how animals interact with their environments, while providing new tools for conservation and management. Chapter 4: Reproduction and Mating Systems. Reproductive behavior encompasses the complex array of strategies that animals have evolved to find mates, compete for reproductive opportunities, and successfully produce offspring that can survive and reproduce in subsequent generations. The diversity of mating systems found in nature reflects the different solutions that species have evolved to address fundamental challenges including mate location, mate choice, offspring care, and genetic diversity maintenance. Understanding reproductive strategies provides crucial insights into evolutionary processes, while revealing the sophisticated behavioral and physiological mechanisms that ensure species persistence across generations. Sexual selection, first described by Charles Darwin, operates through two primary mechanisms that shape the evolution of reproductive behaviors and characteristics. Intrasexual selection involves competition between members of the same sex, typically males, for access to mates or resources that attract mates. This process has led to the evolution of elaborate weapons such as antlers and horns, competitive displays, and aggressive behaviors that determine which individuals gain mating opportunities. Intersexual selection, also known as mate choice, involves individuals of one sex, typically females, selecting mates based on particular characteristics or behaviors that may indicate genetic quality, parental ability, or other desirable traits. The fundamental asymmetry between male and female reproductive biology creates different evolutionary pressures that shape sex-specific behaviors and strategies. In most species, females invest more heavily in individual offspring through the production of large, nutrient-rich eggs and often extensive parental care, while males typically produce numerous small sperm with minimal additional investment. This difference leads to predictions that females should be choosier about mates, while males should compete more intensively for mating opportunities. Mating systems vary tremendously across animal species, ranging from strict monogamy where individuals form exclusive pair bonds, to extreme polygamy where successful individuals mate with numerous partners. Monogamous systems often evolve when both parents are required for successful offspring rearing, or when mate guarding is effective in ensuring paternity. Social monogamy, where pairs remain together for extended periods, may coexist with genetic polyandry if extra pair copulations occur, highlighting the complexity of actual reproductive patterns. Polygamous mating systems include polygyny, where males mate with multiple females, and polyandry, where females mate with multiple males. Polygynous systems often evolve when males can monopolize multiple females, or when environmental conditions allow some males to control territories or resources that attract numerous females. Polyandrous systems are less common, but may evolve when male parental care is particularly valuable, or when females can benefit from mating with multiple males to ensure fertilization or gain genetic diversity. Courtship behaviors serve multiple functions including species recognition, mate assessment, and synchronization of reproductive physiology. These elaborate displays often involve complex sequences of visual, acoustic, or chemical signals that demonstrate male quality while stimulating female reproductive readiness. The peacock spider's intricate courtship dance, complete with colorful displays and rhythmic movements, exemplifies how sexual selection can produce extraordinary behavioral repertoires devoted entirely to attracting mates. Many animals engage in extensive mate choice behaviors that involve evaluation of potential partners based on various criteria including physical condition, territorial quality, genetic compatibility, and behavioral displays. Female choice can be based on direct benefits such as nuptial gifts, territory quality, or parental care, or on indirect benefits such as good genes that may improve offspring survival and reproductive success. The complexity of mate choice decisions reveals sophisticated cognitive abilities and information processing capacities. Alternative reproductive strategies have evolved in many species as ways for individuals to achieve reproductive success when they cannot compete successfully through conventional means. Sneaker males may attempt to intercept matings between dominant males and females, while satellite males may mimic females to approach mating pairs undetected. These alternative strategies often involve different developmental pathways and can be maintained in populations through frequency-dependent selection or conditional strategies based on individual condition or environmental factors. Parental care behaviors range from simple nest construction and egg guarding to complex provisioning and protection of offspring that may continue for months or years after birth. The evolution of parental care involves trade-offs between current reproductive effort and future reproductive opportunities, with the optimal level of care depending on factors including offspring survival benefits, parental survival costs, and the likelihood of future breeding opportunities. Biparental care systems require coordination between males and females in providing for offspring needs, while managing potential conflicts over the level and type of care provided by each parent. These systems often involve complex negotiations and signaling between partners to maintain cooperation, while ensuring that neither parent is exploited by the other. Divorce and mate switching may occur when individuals can improve their reproductive prospects by finding new partners. Sexual conflict arises when the reproductive interests of males and females diverge, leading to evolutionary arms races between the sexes. Males may evolve traits that increase their mating success at the expense of female fitness, while females evolve counter-adaptations to resist male manipulation or exploitation. These conflicts can drive rapid evolutionary change and may contribute to reproductive isolation and speciation processes. Sperm competition occurs when females mate with multiple males, creating competition between the sperm of different males for fertilization opportunities. This process has led to the evolution of diverse male adaptations, including increased sperm production, sperm displacement mechanisms, and mate guarding behaviors that reduce the likelihood of female remating. Females may benefit from sperm competition through increased fertilization rates or the opportunity for cryptic female choice among competing sperm. Reproductive timing and synchronization involve complex physiological and behavioral mechanisms that ensure mating occurs when conditions are optimal for offspring survival. Many species show seasonal breeding patterns that align reproduction with favorable environmental conditions, while others respond to specific environmental cues that predict future resource availability. The ability to time reproduction appropriately is crucial for maximizing reproductive success. Social mating systems in group-living species create additional complexities, including dominance hierarchies that influence mating access, cooperative breeding systems where some individuals help raise offspring they did not produce, and complex alliances that affect reproductive opportunities. These systems demonstrate how social behavior and reproductive behavior are intimately connected and must be understood together. Human activities have increasingly affected animal reproductive systems through habitat fragmentation that disrupts mating encounters, pollution that interferes with chemical communication, and climate change that alters breeding timing and success. Understanding these impacts is crucial for conservation efforts aimed at maintaining viable breeding populations in altered environments. The study of reproductive behavior continues to reveal new complexities, as molecular techniques allow detailed analysis of paternity and genetic relationships, while technological advances enable continuous monitoring of individual behavior and reproductive success. These developments are providing unprecedented insights into the actual mating patterns and reproductive strategies that occur in natural populations, often revealing substantial deviations from predictions based on social behavior alone. Chapter 5: Learning, Memory, and Cognition. Animal cognition encompasses the mental processes that enable animals to acquire, store, and utilize information from their environment, while making decisions that affect their survival and reproductive success. The study of animal learning and memory has revealed remarkable cognitive abilities across diverse animal taxa, challenging traditional assumptions about the uniqueness of human intelligence, while providing insights into the evolutionary origins and adaptive functions of complex mental processes. Understanding animal cognition requires examining both the mechanisms underlying learning and memory, and the ecological contexts in which these abilities have evolved and are expressed. Learning represents a fundamental biological process that allows animals to modify their behavior based on experience, enabling adaptation to environmental variation and unpredictability that cannot be addressed through purely innate responses. The capacity for learning provides animals with behavioral flexibility that can be crucial for survival in dynamic environments where fixed behavioral programs might be inadequate or maladaptive. Different types of learning have evolved to address different challenges, from simple habituation that reduces responses to irrelevant stimuli, to complex problem-solving abilities that enable animals to develop novel solutions to unprecedented problems. Classical conditioning, first systematically studied by Ivan Pavlov, involves learning associations between environmental stimuli and biologically significant events. This form of learning allows animals to anticipate important events based on predictive cues, enabling them to prepare for food availability, predator presence, or other significant circumstances. The widespread occurrence of classical conditioning across animal taxa demonstrates its fundamental importance for survival, while variations in conditioning abilities reflect different ecological pressures and cognitive requirements. Operant conditioning involves learning associations between behaviors and their consequences, allowing animals to increase behaviors that produce beneficial outcomes while decreasing behaviors that result in negative consequences. This form of learning enables animals to discover effective strategies for obtaining food, avoiding predators, and navigating social interactions. The principles of operant conditioning have been documented across diverse species and provide the foundation for understanding how animals learn to solve complex problems through trial and error. Spatial learning and navigation involves sophisticated cognitive processes that enable animals to move efficiently through their environments while maintaining knowledge of resource locations, territorial boundaries, and safe refuges. Many animals demonstrate remarkable spatial memory abilities, with some species capable of remembering hundreds or thousands of cache locations or navigating across vast distances using complex environmental cues. The hippocampus plays a crucial role in spatial learning in vertebrates, with comparative studies revealing interesting relationships between hippocampal size and spatial memory demands. Social learning occurs when animals acquire new behaviors or information through observation of or interaction with other individuals, enabling rapid transmission of adaptive behaviors without the costs and risks associated with individual trial and error learning. Cultural transmission through social learning has been documented in numerous species, from tool use techniques in chimpanzees and dolphins to foraging strategies in birds and migration routes in whales. These cultural phenomena demonstrate that behavior can be inherited through social as well as genetic mechanisms. Imitation and teaching represent sophisticated forms of social learning that involve complex cognitive processes, including attention to others' actions, understanding of causality, and intentional information transfer. True imitation requires animals to copy the specific actions of others rather than simply achieving the same results through different means, while teaching involves individuals actively facilitating learning in others, often at some cost to themselves. Both abilities have been documented in several species, and may represent important precursors to human cultural evolution. Problem-solving abilities enable animals to develop novel solutions to challenges that they have not previously encountered, demonstrating cognitive flexibility and creative thinking that go beyond simple associative learning. Many animals show remarkable problem-solving skills, from corvids using tools to access food to elephants cooperating to solve complex puzzles. These abilities often involve insight learning, where solutions appear suddenly without obvious trial and error processes, suggesting sophisticated mental representations and reasoning abilities. Working memory and executive function involve the temporary storage and manipulation of information needed to guide ongoing behavior, enabling animals to maintain goal-directed behavior despite distractions and competing demands. These cognitive abilities are crucial for complex behaviors, including foraging in multiple locations, social interactions involving several individuals, and multi-step problem-solving tasks. Research on working memory in animals has revealed both similarities to and differences from human cognitive processes. Numerical cognition has been demonstrated in numerous animal species, with many animals showing abilities to discriminate between different quantities, perform simple arithmetic operations, and use numerical information to guide decision-making. These abilities suggest that mathematical thinking has deep evolutionary roots and may serve important functions in contexts including foraging decisions, territorial assessment, and social interactions. The study of numerical cognition in animals provides insights into the biological foundations of mathematical abilities. Metacognition, the ability to monitor and control one's own cognitive processes, has been investigated in several animal species with intriguing results. Some animals demonstrate uncertainty monitoring, showing hesitation or seeking additional information when facing difficult decisions, while others show memory monitoring, accurately assessing their own knowledge and adjusting behavior accordingly. These abilities, suggests that some animals may have awareness of their own mental states and cognitive limitations. Communication and language-like abilities in animals have been extensively studied, revealing sophisticated information transmission systems that share some features with human language while lacking others. Primate language studies have demonstrated that some apes can learn to use symbolic communication systems with considerable complexity, while research on natural communication systems has revealed remarkable complexity in the vocalizations of dolphins, elephants, and various bird species. The neural basis of animal cognition involves complex brain systems that process sensory information, store memories, and generate behavioral responses. Comparative neuroscience has revealed both conserved mechanisms that operate across diverse taxa and specialized adaptations that reflect particular cognitive demands. The relationship between brain size, brain organization, and cognitive abilities continues to be an active area of research with important implications for understanding the evolution of intelligence. Cognitive ecology examines how cognitive abilities have evolved in response to specific environmental challenges and how these abilities function in natural contexts. This approach recognizes that cognitive abilities are adaptations that have been shaped by natural selection to solve particular problems, and that understanding cognition requires consideration of the ecological context in which it operates. Different species face different cognitive challenges based on their ecological niches, leading to diverse cognitive specializations. Individual differences in cognitive abilities have become increasingly recognized as important sources of variation within species, with some individuals consistently outperforming others on cognitive tasks. These differences may reflect genetic variation, developmental experiences, or environmental factors, and can have important consequences for individual fitness and population dynamics. Understanding cognitive variation is crucial for predicting how animals will respond to environmental changes and human impacts. The study of animal cognition has important implications for animal welfare, conservation, and our understanding of human cognitive evolution. Recognition of sophisticated cognitive abilities in animals has led to increased concern about the ethical treatment of animals in research, captivity, and wild populations. Understanding how animals perceive and respond to environmental changes is crucial for developing effective conservation strategies, while comparative studies of cognition provide insights into the evolutionary origins of human mental abilities and the factors that contribute to the evolution of intelligence. Comparative anatomy, examining the anatomy of different species. By Ray Arters. Chapter 1: The Foundation of Form. The study of comparative anatomy stands as one of biology's most revealing disciplines, offering profound insights into the relationships between organisms and the evolutionary processes that have shaped life on Earth. By examining the structural similarities and differences across species, we uncover the fundamental principles that govern biological design and understand how form follows function in the natural world. Comparative anatomy emerged as a formal scientific discipline in the 18th and 19th centuries, though humans have always been curious about the similarities between their own bodies and those of other animals. The French naturalist Georges Cuvier is often credited as the father of comparative anatomy, establishing the principle that anatomical structures must be understood in relation to their function and the organism's way of life. His work laid the groundwork for understanding that anatomical features don't exist in isolation, but are part of integrated systems that enable survival in specific environments. The discipline gained tremendous significance with Charles Darwin's theory of evolution, as anatomical similarities between species provided compelling evidence for common ancestry. Homologous structures, those sharing the same fundamental blueprint despite serving different functions, became powerful indicators of evolutionary relationships. The pentadactyl limb, for instance, appears across vertebrates from bats to whales to humans, each modified for its specific purpose, yet retaining the same basic 5-digit pattern inherited from a common ancestor. Modern comparative anatomy extends far beyond simple structural comparisons. Today's anatomists employ sophisticated imaging techniques, molecular tools, and computational methods to explore anatomy at multiple scales, from gross morphology down to cellular and even molecular levels. Magnetic resonance imaging allows researchers to examine internal structures in living animals without dissection, while electron microscopy reveals ultrastructural details that connect form to function at the cellular level. The field has also embraced developmental approaches, recognizing that understanding how structures form during embryonic development often reveals more about evolutionary relationships than adult anatomy alone. The study of evolutionary developmental biology, or EVO-DEVO, has shown that similar developmental programs can produce remarkably different adult forms, while different developmental pathways can sometimes converge on similar structural solutions. Environmental pressures play a crucial role in shaping anatomical diversity. The principle of convergent evolution demonstrates how unrelated organisms can develop remarkably similar structures when facing comparable environmental challenges. The streamlined body forms of sharks, dolphins, and ichthyosaurs exemplify this phenomenon, each group independently evolving hydrodynamic shapes optimized for aquatic locomotion despite their vastly different evolutionary origins. The relationship between structure and function represents one of comparative anatomy's core principles. Every anatomical feature exists because it serves, or once served, a specific purpose in the organism's survival and reproduction. This functional approach helps explain why certain structural patterns appear repeatedly across different groups of organisms. The evolution of flight, for example, has occurred independently in insects, birds, bats, and extinct reptiles, each group developing unique solutions to the engineering challenges of powered flight while adhering to the same basic aerodynamic principles. Scale effects profoundly influence anatomical design across different-sized organisms. Small animals face different physical constraints than large ones, leading to predictable changes in body proportions and internal architecture. Surface area to volume ratios affect everything from metabolic rates to heat regulation, explaining why small mammals typically have higher metabolic rates and more elaborate circulatory systems relative to their body size than their larger relatives. The integration of systems represents another fundamental aspect of comparative anatomy. No organ system functions in isolation. Cardiovascular, respiratory, nervous, and skeletal systems must work together harmoniously. Changes in one system often necessitate coordinated changes in others, creating evolutionary constraints that channel anatomical diversity along certain paths, while making other combinations unlikely or impossible. Comparative anatomy also reveals the constraints imposed by evolutionary history. Not every theoretically optimal design is achievable, because evolution works with existing structures rather than designing from scratch. The recurrent laryngeal nerve in mammals, which takes a lengthy detour around the aortic arch, even in giraffes, exemplifies how evolutionary history can result in seemingly inefficient anatomical arrangements that persist because the cost of reorganization exceeds the benefit of optimization. Chapter 2: Skeletal Systems Across the Animal Kingdom. The skeletal system provides the structural framework that defines an organism's shape, protects vital organs, and enables movement. Across the animal kingdom, evolution has produced an extraordinary diversity of skeletal solutions, each representing a unique response to environmental pressures and functional demands. Understanding these variations illuminates fundamental principles of biomechanics and reveals how different evolutionary lineages have solved similar structural challenges. Vertebrate skeletons represent perhaps the most familiar skeletal system, characterized by an internal framework of bone and cartilage that grows with the organism throughout its life. The vertebrate skeleton's modular design, built around the vertebral column, provides both structural integrity and flexibility. This segmented approach allows for complex movements, while maintaining the structural support necessary for large body sizes. The evolution of bone itself represents a remarkable innovation, combining the strength of mineral components with the flexibility of organic matrices to create a material that can withstand both tension and compression, while remaining lightweight enough for efficient locomotion. The transition from aquatic to terrestrial environments drove many of the most significant innovations in vertebrate skeletal design. Aquatic vertebrates benefit from water's buoyant support, allowing for more gracile skeletal structures, while terrestrial animals require robust frameworks capable of supporting their full body weight against gravity. This transition is clearly visible in the fossil record, showing the gradual strengthening of limb bones and the development of more elaborate joint systems as vertebrates colonized land environments. Bird skeletons represent one of evolution's most elegant solutions to the challenge of powered flight. Every aspect of avian skeletal anatomy reflects the demands of aerial locomotion, from the fusion of bones to reduce weight while maintaining strength, to the evolution of pneumatic bones filled with air spaces that further reduce mass without compromising structural integrity. The bird sternum has evolved into a prominent keel that provides attachment sites for the massive flight muscles, while the wing bones have been modified into a highly efficient airfoil support system. Mammalian skeletal diversity reflects the group's remarkable ecological success across virtually every terrestrial habitat. Cursorial mammals like horses have evolved elongated limb bones and reduced digit numbers to maximize stride efficiency, while fossorial species such as moles have developed powerful forelimbs with enlarged attachment sites for digging muscles. Aquatic mammals present fascinating examples of skeletal modification, with whales showing extreme limb reduction and vertebral column modifications that enable their undulatory swimming style. Arthropod exoskeletons represent a fundamentally different approach to structural support, relying on external armor-like coverings that must be periodically shed and replaced as the animal grows. This exoskeletal system provides excellent protection and precise control of movement through its system of joints and muscle attachments, but it also imposes constraints on maximum body size due to the scaling relationships between surface area and volume. The arthropod exoskeleton's segmented design allows for remarkable diversity in body plans, from the streamlined forms of aquatic crustaceans to the specialized appendages of terrestrial insects. The compound nature of arthropod exoskeletons, composed primarily of chitin and proteins, creates a material that can be selectively hardened in some areas while remaining flexible in others. This allows for the development of specialized structures such as the mandibles of predatory insects or the chelae of crabs, each optimized for specific functions while maintaining integration with the overall body plan. The periodic molting required by exoskeletal growth has also driven the evolution of complex hormonal control systems and behavioral adaptations that protect vulnerable individuals during the molting process. Echinoderm skeletons present yet another solution to structural support, consisting of calcium carbonate plates embedded within the living tissue. This endoskeletal system provides protection while allowing for the unique hydraulic locomotion system that characterizes many echinoderms. The pentaradial symmetry typical of adult echinoderms reflects deep evolutionary constraints, yet this seemingly rigid body plan has been modified to produce the remarkable diversity seen in sea stars, sea urchins, and sea cucumbers. Molluscan shells represent one of nature's most successful protective strategies, with over 100,000 described species employing calcium carbonate shells of extraordinary diversity. From the simple cap-like shells of limpets to the complex chambered shells of nautiluses, Mollusk shells demonstrate how a basic calcium carbonate secretion system can be modified to produce structures serving functions ranging from protection to buoyancy control. The coiled shells of gastropods provide an elegant solution to the problem of increasing internal volume while minimizing surface area, while the bivalve design optimizes protection while allowing for filter feeding. Cnidarian skeletal systems, exemplified by coral reefs, showcase how simple organisms can create some of Earth's largest biological structures. The calcium carbonate skeletons secreted by colonial corals accumulate over thousands of years to form massive reef systems that support extraordinary biodiversity. These skeletal systems demonstrate how individual organisms can collectively engineer entire ecosystems through their structural contributions. The study of fossil skeletons provides invaluable insights into evolutionary transitions and extinct body plans. The Cambrian explosion preserved examples of experimental skeletal designs that have no modern equivalents, suggesting that early animal evolution explored a much broader range of structural solutions than are represented in contemporary fauna. These fossil forms help us understand the constraints and opportunities that have shaped skeletal evolution throughout Earth's history. Biomineralization processes underlying skeletal formation represent one of biology's most sophisticated manufacturing systems. The precise control of crystal nucleation, growth and orientation allows organisms to create materials with properties that often exceed those of purely inorganic systems. Understanding these processes has applications beyond comparative anatomy, inspiring the development of new materials and manufacturing techniques based on biological principles. Chapter 3: Circulatory Systems and Fluid Transport. The evolution of circulatory systems represents one of biology's most crucial innovations, enabling the efficient transport of nutrients, gases, waste products, and signaling molecules throughout increasingly complex organisms. As multicellular life evolved beyond the simple diffusion-based transport possible in small organisms, natural selection favored increasingly sophisticated circulatory arrangements that could service the metabolic demands of larger, more active animals. The simplest multicellular organisms rely entirely on diffusion for internal transport, a strategy that works effectively only when all cells are within a few cell layers of the external environment. This constraint explains why organisms like cnidarians and flatworms maintain relatively thin body plans despite their multicellular complexity. However, as evolutionary pressures favored larger body sizes and more active lifestyles, the limitations of diffusion-based transport necessitated the development of dedicated circulatory systems. Open circulatory systems represent the first major evolutionary solution to long-distance transport challenges. Found in arthropods and most mollusks, these systems pump hemolymph through a series of vessels that empty into large body cavities called hemocoels, where the fluid directly bathes the tissues before returning to the heart. This design provides adequate transport for many functions, while remaining relatively simple and energy efficient. Arthropod hearts, despite their apparent simplicity, demonstrate remarkable sophistication in their valve systems and neural control mechanisms that optimize flow patterns for different physiological demands. The evolution of closed circulatory systems marked a revolutionary advance in transport efficiency. By confining blood within a continuous network of vessels, these systems achieve higher pressures and more precise control of flow distribution. This design innovation enabled the evolution of larger, more metabolically active organisms, while providing the foundation for sophisticated physiological regulation. The earthworm's multiple hearts illustrate how even relatively simple closed systems can achieve effective circulation through anatomical multiplication of pumping centers. Vertebrate circulatory systems showcase the most advanced examples of closed circulation, with hearts that have evolved from simple tube-like structures to complex multi-chambered organs capable of generating high pressures while maintaining separate pulmonary and systemic circuits. The evolutionary progression from the single-circuit system of fish to the double-circuit systems of birds and mammals reflects the changing demands imposed by terrestrial life and the evolution of air-breathing respiratory systems. Fish circulatory systems demonstrate elegant simplicity in their single-circuit design, where blood flows from the heart to the gills for oxygenation, then directly to body tissues before returning to the heart. This arrangement works efficiently in aquatic environments, where gill ventilation can extract sufficient oxygen from water, but it becomes limiting in air-breathing vertebrates where the resistance of lung circulation would significantly reduce systemic blood pressure if not separated into a distinct circuit. Amphibian hearts represent a transitional stage in circulatory evolution, with their three-chambered design, allowing for partial separation of oxygenated and deoxygenated blood while maintaining a single ventricle. This arrangement supports the dual aquatic and terrestrial lifestyle of many amphibians, providing flexibility in circulation patterns that can be adjusted based on whether gas exchange is occurring primarily through lungs, skin, or gills. The four-chambered hearts of birds and mammals represent the pinnacle of vertebrate circulatory evolution, achieving complete separation of pulmonary and systemic circuits. This design enables high systemic blood pressures necessary for efficient perfusion of large bodies, while maintaining lower pulmonary pressures that prevent fluid accumulation in the lungs. The independent evolutionary achievement of four-chambered hearts in birds and mammals demonstrates convergent evolution, driven by the similar physiological demands of high metabolism, active lifestyles. Specialized circulatory adaptations reflect the diverse ecological niches occupied by different animals. Diving mammals have evolved remarkable modifications including enlarged blood volumes, increased oxygen storage capacity, and sophisticated cardiovascular adjustments that enable extended breath-holding periods. The diving response involves selective vasoconstriction that prioritizes blood flow to essential organs while reducing circulation to less critical tissues. High-altitude adaptations in various mammals illustrate how circulatory systems can respond to environmental challenges through both anatomical and physiological modifications. Andean vicuñas and Tibetan yaks have evolved increased red blood cell counts, enhanced oxygen carrying capacity, and modified heart structures that enable efficient circulation despite reduced atmospheric oxygen levels. The lymphatic system represents a crucial but often overlooked component of vertebrate circulation, serving essential roles in fluid balance, immune function, and fat absorption. This secondary circulatory network demonstrates how evolutionary solutions often involve the development of complementary systems that work together to address complex physiological challenges. The lymphatic system's valve structure and pumping mechanisms show remarkable convergence with venous circulation while serving distinct functional roles. Invertebrate circulatory systems display extraordinary diversity in their approaches to fluid transport. Cephalopod mollusks have evolved closed circulatory systems with multiple hearts that rival vertebrate systems in their sophistication, supporting the high metabolic demands of their active predatory lifestyle. The presence of both systemic and gill hearts in octopuses and squid demonstrates how evolutionary pressures can drive the independent evolution of complex circulatory arrangements. Insect circulatory systems present a fascinating alternative approach to fluid transport, with their open system supporting extremely high metabolic rates through integration with the tracheal respiratory system. The insect heart's segmented structure and sophisticated valve systems enable efficient hemolymph circulation, while the separate tracheal system handles gas transport directly to tissues, eliminating the circulatory system's respiratory function seen in other animals. The study of circulatory system evolution provides insights into fundamental principles of fluid mechanics and physiological optimization. The relationship between heart structure, vessel architecture, and flow dynamics demonstrates how evolutionary processes have repeatedly discovered optimal solutions to transport challenges, often converging on similar designs despite independent evolutionary origins. Chapter 4: Respiratory Systems and Gas Exchange. The diversity of respiratory systems across the animal kingdom reflects the fundamental challenge of obtaining sufficient oxygen, while eliminating carbon dioxide in environments ranging from deep ocean trenches to high mountain peaks. The evolution of gas exchange mechanisms has proceeded along multiple independent pathways, each representing a unique solution to the physical and chemical constraints imposed by different respiratory media and metabolic demands. Simple diffusion across cell membranes suffices for gas exchange in the smallest organisms, where short diffusion distances and high surface area-to-volume ratios ensure adequate oxygen delivery to all cells. However, as organisms evolved larger sizes and higher metabolic rates, natural selection favored the development of specialized respiratory surfaces with enhanced surface areas and optimized transport mechanisms. The transition from diffusion-based to convection-based respiratory systems represents one of the most significant innovations in animal evolution. Aquatic respiratory systems face the unique challenges of extracting dissolved oxygen from water, a medium that contains roughly 30 times less oxygen than air and is nearly 1,000 times denser. Fish gills represent one of evolution's most elegant solutions to aquatic gas exchange, employing a countercurrent flow arrangement that maximizes oxygen extraction efficiency. The gill structure demonstrates remarkable architectural sophistication, with multiple levels of branching that create enormous surface areas within compact spaces. The countercurrent flow principle underlying gill function illustrates how physical laws constrain and direct evolutionary solutions. By flowing water and blood in opposite directions across the respiratory surface, fish achieve oxygen extraction efficiencies approaching 80 to 85%, far exceeding what would be possible with concurrent flow arrangements. This principle has evolved independently in various aquatic animals, demonstrating convergent evolution driven by the mathematical optimization of gas exchange. Insect respiratory systems represent a radically different approach to gas exchange, bypassing circulatory transport entirely through direct delivery of air to tissues via branching tracheal networks. This system enables extraordinary metabolic rates and supports the rapid flight metabolism of insects, but it also imposes constraints on maximum body size due to the scaling limitations of diffusion-based transport within the tracheal system. The evolution of active ventilation mechanisms in larger insects shows how respiratory systems can be modified to overcome initial design constraints. The tracheal system's spiracle-controlled air entry points demonstrate sophisticated regulation mechanisms that balance gas exchange needs with water conservation requirements. Desert insects have evolved particularly elaborate spiracle control systems that minimize water loss while maintaining adequate oxygen supply, illustrating how respiratory systems must integrate with overall water balance strategies. Vertebrate lungs showcase remarkable diversity in their structural approaches to air breathing, from the simple sac-like lungs of amphibians to the highly complex parabronchial systems of birds. Mammalian lungs employ a dead-end alveolar design that achieves efficient gas exchange through enormous surface areas created by millions of tiny alveolar sacs. The branching pattern of mammalian airways demonstrates fractal geometry that optimizes air distribution while minimizing the energy costs of breathing. Avian respiratory systems represent perhaps the most sophisticated gas exchange mechanisms in the animal kingdom, employing a flow-through design that maintains unidirectional airflow through the gas exchange surfaces. This arrangement enables birds to extract more oxygen from each breath than mammals can achieve, supporting the extraordinarily high metabolic demands of powered flight. The avian respiratory system's integration with hollow bones creates a distributed gas exchange network that extends throughout much of the body. The evolution of air breathing required numerous coordinated changes beyond respiratory surface modifications. The development of mechanisms for air movement, from the simple buccal pumping of amphibians to the complex muscular arrangements driving mammalian ventilation, shows how respiratory evolution involves entire systems rather than isolated structures. The evolution of the diaphragm in mammals represents a key innovation that enabled efficient ventilation of large lung volumes. Diving adaptations reveal how respiratory systems can be modified for extreme environmental conditions. Marine mammals have evolved numerous specializations, including collapsible lungs, enhanced oxygen storage in blood and muscle, and sophisticated diving reflexes that optimize oxygen utilization during extended breath-holding periods. The sperm whale's ability to dive to depths exceeding 2,000 meters demonstrates the remarkable physiological modifications possible within vertebrate respiratory systems. High-altitude respiratory adaptations illustrate another dimension of respiratory system plasticity. Animals living at extreme elevations face reduced atmospheric pressure and oxygen availability, driving evolutionary adaptations including increased lung surface areas, enhanced oxygen carrying capacity, and modified ventilation patterns. The bar-headed goose's ability to fly over Mount Everest showcases respiratory modifications that enable function in environments that challenge the limits of vertebrate physiology. Some of the most remarkable respiratory adaptations occur in animals that must function in multiple respiratory environments. Amphibians typically employ combinations of lung, skin, and gill respiration at different life stages or environmental conditions, requiring complex physiological coordination between different gas exchange surfaces. The respiratory flexibility of amphibians illustrates how evolutionary solutions often involve multiple complementary systems rather than single optimal designs. Air-breathing aquatic animals face the unique challenge of obtaining oxygen from air while living in water, leading to fascinating convergent solutions across unrelated groups. Whales, sea turtles, and aquatic insects have all evolved behavioral and physiological adaptations for air breathing in aquatic environments, including modified breathing patterns, enhanced oxygen storage, and specialized surfacing behaviors. The study of respiratory pigments reveals another dimension of respiratory system diversity. While most vertebrates rely on hemoglobin for oxygen transport, various invertebrate groups have evolved alternative respiratory pigments, including hemocyanin in arthropods and mollusks, and chlorocruorin in some polychaete worms. These different pigments demonstrate how evolution has explored multiple chemical solutions to oxygen transport challenges. Plant respiratory systems, though often overlooked in animal-focused discussions, provide important comparative insights into gas exchange evolution. The stomatal control systems of plants demonstrate sophisticated regulatory mechanisms that balance gas exchange with water conservation, employing guard cell mechanisms that parallel the spiracle control systems of insects, despite their independent evolutionary origins. Chapter 5: Sensory Systems and Environmental Interface. The evolution of sensory systems represents one of nature's most remarkable achievements in biological engineering, enabling organisms to detect, process, and respond to an enormous range of environmental stimuli. From the simple light-sensitive eye spots of single-celled organisms to the sophisticated compound eyes of arthropods and the camera-like eyes of vertebrates, sensory evolution demonstrates how natural selection can craft exquisitely sensitive detection systems that gather information crucial for survival and reproduction. Vision systems showcase perhaps the most dramatic diversity in sensory evolution, with eyes having evolved independently at least 40 times across different animal lineages. This remarkable convergence reflects the tremendous survival advantage conferred by the ability to detect light and form images of the surrounding environment. The physics of light detection and image formation have imposed similar constraints across all evolutionary lineages, yet the solutions developed show extraordinary creativity in their structural approaches. Compound eyes, found in arthropods and some other invertebrates, employ thousands of individual optical units called ommastия, each functioning as a separate photoreceptor with its own lens system. This design excels at motion detection and provides an extremely wide field of view, making it ideal for detecting predators and prey in complex environments. The compound eye's modular construction also allows for regional specialization, with some areas optimized for high resolution, while others prioritize motion sensitivity or light-gathering ability. Camera-like eyes, exemplified by vertebrates and cephalopod mollusks, focus light through a single lens onto a photoreceptive surface, creating detailed images similar to those formed by artificial cameras. The independent evolution of camera eyes in vertebrates and cephalopods represents one of evolution's most striking examples of convergent design, with both groups developing remarkably similar solutions to the challenges of image formation, focus adjustment, and light regulation. The diversity of photoreceptive pigments across different animals reveals how evolution has exploited various molecular approaches to light detection. While many animals rely on rhodopsin-based visual pigments, others have evolved alternative systems that extend their spectral sensitivity into ultraviolet or infrared ranges invisible to human perception. The tetrachromatic vision of many birds, incorporating 4 types of color receptors instead of the 3 found in most mammals, demonstrates how evolutionary modifications can enhance color discrimination capabilities. Echolocation represents one of the most sophisticated sensory innovations in the animal kingdom, independently evolved by bats, dolphins, and some other mammals. These biological sonar systems generate high-frequency sounds and analyze the returning echoes to create detailed 3-dimensional maps of their environment. The neural processing required for echolocation rivals the complexity of any sensory system, involving precise temporal analysis and sophisticated signal processing that enables navigation and hunting in complete darkness. The evolution of echolocation required coordinated changes in multiple organ systems, including specialized sound production mechanisms, enhanced auditory processing capabilities, and modified brain structures for echo analysis. Bat echolocation shows remarkable diversity in its implementations, from the constant frequency calls of horseshoe bats that excel at detecting fluttering prey, to the frequency-modulated calls of Vespertilian ID bats that provide detailed information about object texture and distance. Mechanical sensory systems detect physical forces such as touch, pressure, vibration, and acceleration, providing crucial information about the physical environment and the organism's position within it. The lateral line system of fish and aquatic amphibians represents a unique mechanosensory adaptation that detects water movements and pressure changes, enabling these animals to sense nearby objects, currents, and the movements of other animals through hydrodynamic disturbances. The evolution of hearing demonstrates how mechanical sensory systems can be elaborated into sophisticated sound detection mechanisms. The mammalian ear's 3-bone middle ear system, evolved from reptilian jawbones, shows how evolutionary repurposing can create entirely new functional capabilities. The cochlear's frequency analysis capabilities, achieved through mechanical tuning of different regions to specific frequencies, demonstrates how biological systems can perform complex signal processing using purely mechanical principles. Balance and spatial orientation systems, including the vertebrate vestibular system and arthropod statocysts, employ mechanical principles to detect gravity and acceleration. These systems are crucial for coordinated movement and spatial navigation, yet they show remarkable diversity in their structural implementations across different animal groups. The semicircular canal system of vertebrates provides 3-dimensional acceleration detection through fluid-filled tubes oriented in perpendicular planes. Chemical sensory systems encompassing taste and smell represent some of the most ancient sensory modalities, with roots extending back to the earliest cellular life. The molecular recognition mechanisms underlying chemical sensation demonstrate extraordinary specificity, with individual receptor proteins capable of distinguishing between molecules that differ by only a single atom. The diversity of chemical receptors across species reflects their different ecological niches and the chemical environments they must navigate. Olfactory systems show remarkable sensitivity, with some animals capable of detecting individual molecules among billions of others. The elaborate antennae of many insects provide enormous surface areas for chemical reception, while the complex turbinate bones in mammalian noses create turbulent airflow that enhances chemical detection. Salmon navigation using chemical cues demonstrates how olfactory systems can provide precise spatial information across vast distances. Electroreception, found in various aquatic animals including sharks, rays and some bony fish, represents a sensory modality completely absent in terrestrial vertebrates. The ampullae of Lorenzini in sharks can detect electrical fields as weak as a few nanovolts, enabling these predators to locate prey through the bioelectrical activity of living tissues. Some fish have evolved active electroreception, generating their own electrical fields and detecting distortions caused by nearby objects. Magnetic field detection, while still incompletely understood, appears to provide navigational information for many migratory animals. The mechanisms underlying magnetoreception likely involve specialized cells containing magnetite crystals or photochemical processes involving cryptochrome proteins. The ability of various animals to navigate using Earth's magnetic field demonstrates sensory capabilities that extend far beyond human perception. Temperature sensation involves specialized thermoreceptors that detect changes in environmental temperature, crucial for behavioral thermoregulation and habitat selection. Some snakes have evolved highly sensitive infrared detection systems that enable them to locate warm-blooded prey in complete darkness, while Arctic animals have evolved cold-sensing adaptations that help them avoid tissue damage from extreme temperatures. The integration of multiple sensory inputs creates the rich perceptual worlds that animals experience, with different species emphasizing different sensory modalities based on their ecological needs. The sensory biology of different animals reveals that the human sensory experience represents just one narrow slice of the vast array of information available in the natural world, reminding us that each species inhabits its own unique sensory universe, shaped by millions of years of evolutionary refinement.
Chapter 1: The Scope and Origins of Biological Diversity. The diversity of life on Earth represents one of the most awe-inspiring and scientifically fascinating phenomena in the universe, encompassing an estimated 8.7 million species of organisms that inhabit virtually every conceivable environment on our planet. This extraordinary biological richness extends from the deepest ocean trenches to the highest mountain peaks, from scalding hot springs to frozen polar regions, and from the vast expanses of tropical rainforests to the seemingly barren landscapes of deserts and tundra. Each of these environments harbors unique assemblages of organisms that have evolved remarkable adaptations, enabling them to thrive under specific environmental conditions, creating a complex tapestry of life that has been shaped by billions of years of evolutionary innovation and environmental change. Understanding the full scope of biological diversity requires examining life at multiple levels of organization, from the genetic variation present within individual populations to the spectacular array of different species that populate ecosystems worldwide. Genetic diversity represents the foundation upon which all other forms of biological diversity rest, providing the raw material for evolutionary change and adaptation. This genetic variation manifests as differences in DNA sequences among individuals within species, creating the potential for populations to respond to environmental challenges through natural selection and evolutionary adaptation. Species diversity encompasses the remarkable variety of different life forms that have evolved on Earth, each representing a unique solution to the challenges of survival and reproduction in particular environmental contexts. The concept of species itself proves more complex than initially apparent, with biologists recognizing that species boundaries can be fluid and that different species concepts may be appropriate for different groups of organisms. The biological species concept defines species as groups of interbreeding populations that are reproductively isolated from other such groups, while morphological and ecological species concepts focus on structural similarities and ecological roles respectively. Ecosystem diversity reflects the variety of different biological communities and their associated physical environments, ranging from coral reefs and tropical rainforests to grasslands and freshwater lakes. Each ecosystem type represents a unique combination of abiotic factors such as climate, soil type, and water availability, with biotic factors including the species present and their interactions. The diversity of ecosystems on Earth creates numerous distinct habitats that support different assemblages of species, while providing essential services that maintain planetary environmental conditions. The origins of biological diversity trace back to the earliest stages of life's evolution on Earth, beginning approximately 3.5 to 4 billion years ago when the first simple cellular organisms appeared in the primordial oceans. These early life forms were prokaryotic cells lacking the complex internal organization characteristic of modern eukaryotic cells, yet they established the fundamental biochemical and molecular mechanisms that continue to underlie all life processes today. The evolution of photosynthesis in cyanobacteria represents one of the most significant early innovations in the history of life, enabling organisms to harness solar energy while producing oxygen as a byproduct that fundamentally altered Earth's atmospheric composition. The Great Oxidation event that occurred approximately 2.4 billion years ago marked a critical transition in Earth's history when oxygen-producing photosynthesis became widespread enough to dramatically increase atmospheric oxygen concentrations. This environmental change had profound consequences for life on Earth, creating new opportunities for aerobic metabolism while simultaneously driving many anaerobic organisms to extinction or relegating them to oxygen-free environments. The availability of oxygen enabled the evolution of complex multicellular organisms that could achieve much larger sizes and more sophisticated organization than their anaerobic predecessors. The evolution of eukaryotic cells through endosymbiosis represents another major milestone in the diversification of life, enabling the development of complex cellular organization through the incorporation of formerly free-living bacteria as permanent cellular organelles. Mitochondria and chloroplasts retain many characteristics of their bacterial ancestors, including their own genetic systems and reproduction through binary fission, while providing essential functions for cellular energy production and photosynthesis. This endosymbiotic origin of eukaryotic cells enabled the evolution of much more complex organisms, while establishing the foundation for all subsequent diversification of plants, animals, fungi, and protists. The Cambrian explosion that occurred approximately 540 million years ago represents one of the most dramatic episodes of diversification in the history of life, when most major animal phyla first appeared in the fossil record within a relatively short geological time span. This rapid diversification may have been triggered by environmental changes, including increased oxygen levels, the evolution of predator-prey relationships, and the development of hard shells and other defensive structures. The Cambrian period established many of the fundamental body plans that continue to characterize major animal groups today, while demonstrating the potential for rapid evolutionary innovation under appropriate conditions. Mass extinction events have played crucial roles in shaping the diversity of life by eliminating many existing species, while creating opportunities for the radiation of surviving lineages into new ecological niches. The 5 major mass extinction events in Earth's history, including the Permian-Triassic extinction that eliminated approximately 90% of marine species, and the Cretaceous-Paleogene extinction that ended the age of dinosaurs, fundamentally altered the trajectory of life's evolution. These extinction events demonstrate both the vulnerability of life to environmental catastrophes and the remarkable resilience and adaptability of biological systems. Adaptive radiation represents a fundamental mechanism driving the diversification of life, occurring when lineages rapidly evolve to exploit new ecological opportunities or environments. Classic examples include the radiation of Darwin's finches in the Galapagos Islands, where a single ancestral species gave rise to multiple descendant species specialized for different feeding strategies, and the diversification of mammals following the extinction of dinosaurs. These radiations demonstrate how evolutionary processes can rapidly generate new species and ecological relationships when environmental opportunities become available. Geographic isolation has provided a major driver of speciation and diversification throughout the history of life. As populations separated by geographic barriers evolve independently and diverge over time, continental drift and the formation of mountain ranges, ocean basins, and other geographic features have repeatedly isolated populations and promoted speciation. Island biogeography provides particularly clear examples of how geographic isolation promotes diversification, with isolated islands often harboring high levels of endemism where unique species evolve in isolation from mainland populations. Climate change has continuously influenced the diversity and distribution of life throughout Earth's history, driving species migrations, extinctions, and evolutionary adaptations as environmental conditions fluctuated over geological timescales. Ice ages, periods of global warming, and changes in precipitation patterns have repeatedly reshaped ecosystems and species distributions while creating new selective pressures that drove evolutionary innovation. Understanding how past climate changes affected biological diversity provides important insights for predicting how current and future climate change may impact contemporary biodiversity. Coevolution between different species has generated much of the intricate complexity observed in biological communities, as species adapt to interactions with other organisms, including predators, prey, competitors, and mutualistic partners. The evolution of flowering plants and their insect pollinators represents a classic example of coevolution, where reciprocal adaptations have led to remarkable diversity in both flower morphology and pollinator behavior. These coevolutionary relationships demonstrate how species interactions can drive diversification while creating the complex ecological networks that characterize modern ecosystems. Molecular techniques have revolutionized our understanding of biological diversity by revealing previously unknown relationships among organisms while uncovering vast amounts of microbial diversity that was invisible to traditional morphological approaches. DNA sequencing has revealed that prokaryotic diversity far exceeds previous estimates, with many environments harboring hundreds or thousands of microbial species that play crucial roles in ecosystem functioning. Environmental DNA sampling has become a powerful tool for documenting biodiversity in various habitats, while revealing the presence of rare or cryptic species that might otherwise go undetected. The current biodiversity crisis represents an unprecedented threat to the diversity of life, with human activities driving species extinctions at rates far exceeding background extinction rates observed in the fossil record. Habitat destruction, climate change, pollution, invasive species, and overexploitation have combined to create what many scientists consider the 6th mass extinction event in Earth's history. Understanding the patterns and processes that have generated biological diversity throughout Earth's history provides crucial context for efforts to conserve remaining biodiversity and mitigate ongoing extinction threats. Chapter 2: Microbial Diversity and the Invisible Majority. The microbial world represents the most abundant and diverse realm of life on Earth, encompassing an extraordinary array of bacteria, archaea, viruses, and microscopic eukaryotes that collectively dominate virtually every environment on the planet. These microscopic organisms, though largely invisible to the naked eye, constitute the vast majority of life's diversity and biomass, while playing fundamental roles in ecosystem functioning, biogeochemical cycling, and the maintenance of environmental conditions that support all other forms of life. Understanding microbial diversity requires appreciation for both the remarkable metabolic versatility that enables microorganisms to thrive in extreme environments and the sophisticated molecular techniques that have revealed the true scope of microbial richness in recent decades. Bacterial diversity encompasses an enormous range of morphological, physiological, and ecological types that have colonized virtually every habitat on Earth, from the surface of our skin to the depths of ocean sediments and the interior of solid rock formations. The fundamental bacterial cell plan, with its simple prokaryotic organization lacking membrane-bound organelles, has proven remarkably successful and adaptable, enabling bacteria to evolve diverse metabolic strategies and survive under conditions that would be lethal to most other organisms. This success is reflected in the estimated 10 to the power of 30 bacterial cells that exist on Earth, representing a biomass that exceeds all plants and animals combined. The metabolic diversity of bacteria far exceeds that observed in any other group of organisms, with different bacterial species capable of utilizing virtually any organic compound as an energy source while employing diverse strategies for carbon fixation and biosynthesis. Chemosynthetic bacteria can derive energy from the oxidation of inorganic compounds, including hydrogen sulfide, ammonia, and various metals, enabling them to thrive in environments devoid of sunlight, such as deep-sea hydrothermal vents and underground caves. Photosynthetic bacteria include both oxygen-producing, cyanobacteria that perform plant-like photosynthesis, and diverse groups of anoxygenic photosynthetic bacteria that use different photosynthetic pathways. Extremophilic bacteria demonstrate the remarkable ability of life to adapt to conditions that were long considered incompatible with biological processes, including temperatures exceeding 100 degrees Celsius, pH levels below 1 or above 12, salt concentrations approaching saturation, and radiation levels that would quickly kill most other organisms. Thermophilic bacteria thrive in hot springs and hydrothermal vents, while psychrophilic bacteria inhabit permanently frozen environments, including polar ice and deep ocean waters. Halophilic bacteria flourish in salt lakes and salt mines, while acidophilic bacteria can grow in mine drainage and volcanic hot springs with pH levels comparable to battery acid. Archaea represent a distinct domain of prokaryotic organisms that were initially classified as bacteria, but are now recognized as being as different from bacteria as either group is from eukaryotes. These remarkable microorganisms were first discovered in extreme environments and were initially called archaebacteria, but subsequent research revealed that many archaea inhabit more moderate environments, including soils, sediments, and the human microbiome. Archaea exhibit unique biochemical features, including distinctive cell wall compositions, unusual lipid structures, and novel metabolic pathways that distinguish them from both bacteria and eukaryotes. Methanogenic archaea play crucial roles in global carbon cycling by producing methane in anaerobic environments, including wetlands, sediments, and the digestive systems of ruminant animals. These organisms represent the final step in the anaerobic decomposition of organic matter, converting simple organic compounds and carbon dioxide into methane through unique metabolic pathways found only in archaea. The methane produced by these organisms contributes significantly to atmospheric greenhouse gas concentrations, while serving as an energy source for methanotrophic bacteria in many environments. Viral diversity encompasses an enormous array of obligate intracellular parasites that infect virtually every type of cellular organism, while exhibiting remarkable diversity in structure, genome organization, and replication strategies. Viruses represent the most abundant biological entities on Earth, with marine environments alone containing an estimated 10 to the power of 30 viral particles that play crucial roles in microbial ecology and evolution. The diversity of viral forms includes DNA and RNA viruses, single-stranded and double-stranded genomes, and structures ranging from simple helical and icosahedral forms to complex architectures with multiple protein layers and specialized appendages. Bacteriophages, or viruses that infect bacteria, represent the most abundant and diverse group of viruses, while playing fundamental roles in regulating bacterial populations and driving bacterial evolution through horizontal gene transfer. These viruses exhibit remarkable host specificity, with different phage types specialized for particular bacterial species or even specific strains within species. The predatory pressure exerted by bacteriophages helps maintain bacterial diversity by preventing any single bacterial type from dominating microbial communities while promoting the evolution of defensive mechanisms and phenotypic diversity. Protist diversity encompasses a vast array of eukaryotic microorganisms that represent some of the earliest branches in the eukaryotic tree of life, while exhibiting extraordinary morphological and ecological diversity. These organisms include photosynthetic forms, such as diatoms and dinoflagellates, that contribute significantly to global primary productivity, as well as heterotrophic forms, such as amoebas and ciliates, that play important roles as predators and decomposers in microbial food webs. Many protists exhibit complex life cycles involving multiple morphological forms and reproductive strategies that enable them to respond to changing environmental conditions. Marine phytoplankton represent one of the most ecologically important groups of microorganisms, contributing approximately half of global primary productivity, while forming the base of marine food webs that support virtually all ocean life. Diatoms, with their intricate glass-like cell walls called frustules, represent one of the most diverse groups of phytoplankton and contribute disproportionately to marine productivity and carbon cycling. Coccolithophores produce calcium carbonate plates that contribute to ocean chemistry and sediment formation, while dinoflagellates include both photosynthetic species and heterotrophic predators that can form harmful algal blooms under certain conditions. Soil microbial communities harbor extraordinary diversity that rivals or exceeds that found in any other environment, with a single gram of soil containing thousands of different bacterial species, along with diverse populations of archaea, fungi, and protists. These soil microorganisms play essential roles in nutrient cycling, organic matter decomposition, and plant nutrition, while forming complex ecological networks that influence soil fertility and ecosystem productivity. The rhizosphere, or zone of soil influenced by plant roots, represents a particularly diverse microbial habitat where plant exudates support dense and diverse microbial communities. Symbiotic microorganisms form intimate associations with larger organisms that range from mutualistic relationships, providing benefits to both partners, to parasitic associations where microorganisms benefit at the expense of their hosts. The human microbiome includes trillions of bacterial cells, representing hundreds of species that inhabit various body sites and play important roles in digestion, immune function, and protection against pathogens. Nitrogen-fixing bacteria form mutualistic associations with leguminous plants, providing essential nutrients in exchange for carbohydrates and specialized root environments. Deep subsurface microbial communities exist in groundwater, sediments, and solid rock formations at depths of several kilometers below Earth's surface, representing some of the most extreme and isolated microbial habitats on the planet. These organisms often exhibit extremely slow metabolic rates and may represent some of the longest-lived individual cells known to science, with some estimates suggesting individual cells may survive for thousands or even millions of years in these stable environments. The biomass of deep subsurface microorganisms may rival that of surface life, highlighting the vast scope of microbial diversity in environments that were long considered sterile. Horizontal gene transfer among microorganisms represents a fundamental mechanism driving microbial evolution and diversity, enabling the rapid spread of adaptive traits, including antibiotic resistance, virulence factors, and novel metabolic capabilities. This process occurs through several mechanisms, including transformation, transduction, and conjugation, that enable genetic material to move between distantly related organisms. The frequency of horizontal gene transfer in microbial communities means that microbial evolution can proceed much more rapidly than evolution in organisms that rely primarily on vertical inheritance of genetic material. Microbial ecology has revealed the enormous importance of microorganisms in maintaining ecosystem functioning and global environmental conditions, with microbial processes driving biogeochemical cycles that regulate atmospheric composition, soil fertility, and water quality. Microorganisms are responsible for most nitrogen fixation, denitrification, and nitrification processes that maintain global nitrogen cycling, while also playing crucial roles in carbon, sulfur, and phosphorus cycles. Understanding microbial diversity and ecology has become essential for addressing global environmental challenges, including climate change, pollution, and sustainable agriculture. Cultivation-independent methods have revolutionized our understanding of microbial diversity by revealing that traditional cultivation techniques capture only a tiny fraction of the microorganisms present in most environments. Environmental DNA sequencing and metagenomics approaches can detect and characterize microbial communities without requiring cultivation, revealing vast amounts of previously unknown diversity. These techniques have shown that most microbial species have never been cultured in laboratory conditions, and that microbial diversity far exceeds previous estimates based on culturable organisms. Chapter 3: Plant Diversity and the Conquest of Land. The evolution and diversification of plants represents one of the most significant events in the history of life on Earth, fundamentally transforming terrestrial environments while establishing the foundation for all complex terrestrial ecosystems. The transition from aquatic to terrestrial life required plants to overcome numerous challenges, including desiccation, gravity, ultraviolet radiation, and the absence of structural support provided by water, leading to the evolution of remarkable innovations, including vascular systems, structural support tissues, protective waxy coatings, and sophisticated reproductive strategies. The success of plants in colonizing terrestrial environments enabled the subsequent evolution of terrestrial animal life, while creating the oxygen-rich atmosphere that characterizes our modern world. The origins of land plants can be traced to charophyte green algae that inhabited shallow freshwater environments approximately 500 million years ago, where fluctuating water levels and periodic exposure to air created selective pressures for adaptations that would eventually enable the transition to fully terrestrial life. These ancestral plants face the fundamental challenge of maintaining water balance in environments where desiccation posed a constant threat, leading to the evolution of waxy cuticles that reduce water loss while still permitting gas exchange necessary for photosynthesis. The development of stomata, microscopic pores that can open and close to regulate gas exchange and water loss, represents one of the most important innovations enabling plant success on land. Bryophytes, including mosses, liverworts, and hornworts, represent the earliest diverging lineages of land plants and retain many characteristics that reflect their aquatic ancestry while exhibiting important adaptations for terrestrial life. These small, non-vascular plants lack the specialized conducting tissues found in more advanced plants and remain dependent on external water for reproduction, as their sperm must swim through water films to reach eggs. Despite these limitations, bryophytes have achieved remarkable success in many terrestrial environments, particularly in moist habitats where they can form extensive carpets that stabilize soil and provide habitat for numerous other organisms. The evolution of vascular tissues represents a revolutionary innovation that enabled plants to achieve much larger sizes while efficiently transporting water and nutrients throughout their bodies. Xylem tissue conducts water and dissolved minerals from roots to aerial portions of plants, while phloem tissue transports sugars and other organic compounds from photosynthetic leaves to non-photosynthetic tissues including roots and developing organs. The development of lignin, a complex polymer that provides structural support to cell walls, enabled plants to grow tall and compete effectively for light while maintaining structural integrity against gravitational forces. Ferns and their allies represent the most diverse group of seedless vascular plants, exhibiting remarkable morphological diversity while retaining reproduction through spores rather than seeds. These plants achieved particular success during the Carboniferous period, when extensive fern forests dominated many terrestrial environments, contributing to the formation of coal deposits through the accumulation of organic matter in swampy environments. Modern ferns continue to exhibit impressive diversity, with over 12,000 species occupying habitats ranging from tropical rainforests to temperate woodlands and even aquatic environments. The evolution of seeds represents another major innovation in plant evolution, providing protection and nutrition for developing embryos, while enabling dispersal to new habitats without dependence on external water for fertilization. Seeds contain embryonic plants, along with stored nutrients and protective seed coats that enable survival during unfavorable conditions while facilitating long-distance dispersal. This reproductive strategy proved so successful that seed plants now dominate most terrestrial environments and include the vast majority of plant species. Gymnosperms, or naked seed plants, represent the earliest seed plants and include conifers, cycads, ginkgos, and netophytes that collectively exhibit remarkable adaptations for survival in diverse environments. Conifers have achieved particular success in challenging environments, including boreal forests, montane regions, and Mediterranean climates, where their needle-like leaves and efficient water conservation strategies provide advantages over broad-leaved plants. The longevity of many conifers, with some individuals living for thousands of years, demonstrates the effectiveness of their adaptive strategies while contributing to forest stability and carbon storage. Angiosperms, or flowering plants, represent the most recent major plant group to evolve and have achieved unprecedented diversity with over 300,000 species that dominate most terrestrial environments. The evolution of flowers enabled more efficient and targeted pollination strategies, while the development of fruits provided enhanced seed protection and dispersal mechanisms. The rapid radiation of flowering plants during the Cretaceous period coincided with the diversification of insect pollinators, creating complex coevolutionary relationships that have generated much of the spectacular diversity observed in both flowering plants and insects. Floral diversity reflects the remarkable variety of pollination strategies employed by flowering plants, ranging from wind pollination with small, inconspicuous flowers to elaborate animal-pollinated flowers that offer specific rewards including nectar, pollen, or specialized oils. The evolution of different flower shapes, colors, and scents has enabled flowering plants to attract specific pollinators while avoiding cross-pollination with other species, promoting reproductive isolation and speciation. Some flowering plants have evolved deceptive strategies that attract pollinators without providing rewards, while others have developed highly specialized relationships with particular pollinator species. Fruit diversity encompasses an enormous array of structures that facilitate seed dispersal through various mechanisms, including wind, water, and animal vectors. Fleshy fruits attract animal dispersers with nutritious rewards while protecting seeds during passage through digestive systems, enabling long-distance dispersal and colonization of new habitats. Dry fruits employ various mechanical strategies for seed release, including explosive dehiscence, attachment to animal fur, and specialized structures that facilitate wind dispersal. The diversity of fruit types reflects the variety of dispersal strategies employed by flowering plants to maximize reproductive success. Plant secondary metabolites represent a vast array of chemical compounds that play crucial roles in plant defense, reproduction, and environmental interactions, while contributing significantly to the biochemical diversity of life on Earth. These compounds include alkaloids, terpenoids, phenolics, and numerous other chemical classes that can deter herbivores, inhibit pathogens, attract beneficial organisms, or provide protection against environmental stresses. Many plant secondary metabolites have proven valuable for human applications, including medicines, pesticides, and industrial chemicals, highlighting the practical importance of plant chemical diversity. Adaptive radiations have generated remarkable plant diversity in various environments, with classic examples including the Hawaiian Silversword Alliance that diversified from a single colonizing ancestor into dozens of species adapted to diverse island habitats. Cacti represent another striking example of adaptive radiation, with over 2,000 species exhibiting diverse growth forms and ecological strategies while maintaining common adaptations for water conservation in arid environments. These radiations demonstrate how plant lineages can rapidly diversify when colonizing new environments or when ecological opportunities become available. Convergent evolution has produced remarkable similarities among unrelated plant groups that face similar environmental challenges, demonstrating the constraints imposed by physical and biological factors on plant form and function. Succulent plants have evolved independently in numerous plant families as adaptations for water storage in arid environments, while climbing plants have repeatedly evolved similar strategies for reaching light in forest environments. These convergent adaptations highlight the predictable nature of evolutionary responses to environmental challenges, while demonstrating the multiple pathways available for achieving similar functional outcomes. Epiphytic plants have evolved strategies for living on other plants without parasitizing them, gaining access to light in forest canopies while obtaining water and nutrients from atmospheric sources. These plants include diverse groups such as bromeliads, orchids, and ferns that have colonized tree branches and trunks in tropical and temperate forests worldwide. Epiphytic communities can be extraordinarily diverse and contribute significantly to forest biodiversity, while playing important roles in nutrient cycling and water retention in forest ecosystems. Carnivorous plants represent remarkable examples of convergent evolution, where various plant lineages have independently evolved strategies for capturing and digesting animal prey to supplement nutrient acquisition in nutrient-poor environments. These plants employ diverse trapping mechanisms, including pitfall traps, adhesive traps, snap traps, and suction traps that demonstrate the ingenuity of evolutionary solutions to environmental challenges. The diversity of carnivorous plant strategies reflects the variety of prey types and environmental conditions that have selected for these unusual adaptations. Plant community ecology reveals how different plant species interact to form complex communities that exhibit emergent properties beyond those of individual species. Competition for light, water, and nutrients shapes plant community structure, while facilitation and mutualistic relationships can promote diversity and stability. Succession processes demonstrate how plant communities change over time in response to disturbance and environmental change, with different species dominating at different stages of community development. Understanding plant community dynamics has become increasingly important for conservation and restoration efforts as human activities continue to alter natural plant communities worldwide. Chapter 4: Animal Diversity and Adaptive Strategies. Animal diversity encompasses an extraordinary array of organisms that have evolved countless strategies for survival and reproduction across virtually every environment on Earth, from the deepest ocean trenches to the highest mountain peaks and from polar regions to tropical rainforests. The kingdom Animalia includes organisms ranging from simple sponges with no true tissues to complex vertebrates with sophisticated nervous systems, representing approximately 1.5 million described species, with estimates suggesting that millions more remain undiscovered. This remarkable diversity reflects over 500 million years of evolutionary innovation that has produced solutions to fundamental challenges, including locomotion, feeding, reproduction, and environmental adaptation through an amazing variety of body plans, physiological systems, and behavioral strategies. The origins of animal diversity can be traced to the Precambrian period, when the first multicellular animals evolved from unicellular ancestors, establishing the foundation for all subsequent animal evolution. The transition from single-celled to multicellular organization enabled the evolution of specialized cell types that could work together to perform complex functions impossible for individual cells. Early animals likely resembled modern cnidarians or sponges, with simple body plans consisting of 2 or 3 cell layers that formed tissues specialized for different functions including protection, digestion, and reproduction. Body plan diversity among animals reflects fundamental differences in organization that have major implications for how different animals function and interact with their environments. Radial symmetry, characteristic of cnidarians such as jellyfish and sea anemones, creates body plans organized around a central axis that enables these animals to respond equally well to stimuli from any direction. Bilateral symmetry, found in most other animal groups, creates distinct anterior, posterior, and dorsal ventral axes that enable more sophisticated locomotion and the concentration of sensory organs and nervous system components in anterior regions. Digestive system diversity reflects the enormous variety of feeding strategies employed by different animals, ranging from filter feeding and grazing to predation and parasitism. Sponges employ unique feeding strategies, using specialized cells called choanocytes that create water currents and capture microscopic food particles, while cnidarians use stinging cells called nematocysts to capture prey. More complex animals have evolved sophisticated digestive systems, including specialized regions for food storage, chemical breakdown, absorption, and waste elimination that enable efficient processing of diverse food sources. Respiratory system diversity demonstrates the various solutions that animals have evolved for gas exchange, with different strategies reflecting the constraints imposed by body size, habitat, and metabolic requirements. Aquatic animals employ diverse gill structures that maximize surface area for gas exchange while maintaining efficient water flow, while terrestrial animals have evolved internal respiratory systems, including tracheal systems in insects and lungs in vertebrates. Some animals can absorb oxygen directly through their skin, while others have evolved specialized respiratory pigments that enhance oxygen transport capacity. Circulatory system evolution shows a clear progression from simple diffusion-based transport in small animals to sophisticated closed circulatory systems with multiple chambers and specialized blood vessels in larger, more active animals. Open circulatory systems, found in many invertebrates, provide adequate transport for relatively small animals with low metabolic demands, while closed systems enable the precise control of blood flow and pressure necessary for larger animals with high metabolic requirements. The evolution of different heart designs, from simple tubular hearts to complex 4-chambered hearts, reflects the diverse solutions for pumping blood efficiently through different body plans. Nervous system complexity ranges from simple nerve nets in cnidarians to the sophisticated brains and nervous systems found in vertebrates and some invertebrates, such as cephalopods. The evolution of centralized nervous systems enabled more complex behaviors and faster responses to environmental stimuli, while supporting the development of learning, memory, and complex social behaviors. Different animal groups have evolved remarkably diverse sensory capabilities, including echolocation in bats and dolphins, electrical field detection in sharks and electric fish, and magnetic field sensitivity in many migratory animals. Locomotory diversity reflects the enormous variety of ways that animals move through their environments, with different strategies adapted for terrestrial, aquatic, aerial, and arboreal habitats. Swimming has evolved independently in numerous lineages through various mechanisms, including undulatory body movements, jet propulsion, and specialized appendages such as fins and flippers. Flight has evolved independently in insects, birds, and bats through different mechanisms, while numerous animals have evolved gliding capabilities that enable aerial dispersal without powered flight. Reproductive strategies among animals exhibit remarkable diversity, ranging from simple asexual reproduction through budding or fragmentation to complex sexual reproduction involving elaborate courtship behaviors and parental care. Many animals employ mixed reproductive strategies that include both sexual and asexual reproduction depending on environmental conditions, while others exhibit sequential hermaphroditism where individuals change sex during their lifetimes. The evolution of different mating systems, including monogamy, polygamy, and promiscuity, reflects different solutions to the challenges of finding mates and ensuring reproductive success. Social organization in animals ranges from solitary lifestyles to complex societies with sophisticated division of labor and communication systems. Eusocial insects such as ants, bees, and termites have evolved remarkable colonies where most individuals sacrifice their own reproduction to support the reproductive success of related individuals. Many vertebrates exhibit complex social structures, including dominance hierarchies, cooperative breeding, and cultural transmission of behaviors that enhance survival and reproductive success through group living. Defensive strategies demonstrate the diverse ways that animals protect themselves from predators and other threats, including morphological defenses such as armor, spines, and cryptic coloration, as well as behavioral defenses such as group living, vigilance, and escape responses. Many animals have evolved warning coloration that advertises their toxicity or unpalatability to potential predators, while others employ mimicry to gain protection by resembling dangerous or unpalatable species. Some animals have evolved remarkable regenerative capabilities that enable them to replace lost body parts following predator attacks. Ecological specialization has driven much of animal diversity, as different species have evolved adaptations for exploiting specific resources or habitats. Parasites represent one of the most diverse animal groups, with specialized adaptations for living in or on other organisms, while often exhibiting complex life cycles involving multiple host species. Deep-sea animals have evolved remarkable adaptations for life in high-pressure, low-temperature environments with limited food availability, while cave-dwelling animals often exhibit convergent evolution toward reduced pigmentation and enhanced sensory capabilities. Coevolution between animals and other organisms has generated much of the intricate complexity observed in ecological communities, with predator-prey relationships, plant-herbivore interactions, and mutualistic associations driving reciprocal evolutionary changes. The coevolution of flowering plants and their animal pollinators represents one of the most spectacular examples of such interactions, with both groups exhibiting remarkable diversity that reflects their intimate ecological relationships. Similarly, the evolution of fruit characteristics and seed dispersal by animals demonstrates how mutualistic relationships can drive evolutionary innovation in both partners. Migration represents one of the most remarkable animal behaviors, with numerous species undertaking epic journeys that span continents and oceans to exploit seasonal resources or reach appropriate breeding sites. These migrations require sophisticated navigation abilities and physiological adaptations for sustained travel, while demonstrating the importance of habitat connectivity for animal survival. Arctic terns migrate annually from Arctic to Antarctic regions, while monarch butterflies undertake multi-generational migrations that span thousands of kilometers. Conservation challenges facing animal diversity include habitat destruction, climate change, pollution, overexploitation, and invasive species that threaten many animal populations worldwide. The current rate of animal extinctions far exceeds background extinction rates, leading many scientists to conclude that we are experiencing the sixth mass extinction event in Earth's history. Understanding animal diversity and the ecological relationships that maintain it has become crucial for developing effective conservation strategies and preserving the remarkable heritage of animal life for future generations. Molecular techniques have revolutionized our understanding of animal relationships and diversity, revealing unexpected evolutionary connections while uncovering vast amounts of previously unknown diversity, particularly among microscopic animals and those inhabiting remote or extreme environments. Environmental DNA sampling has become a powerful tool for documenting animal diversity in various habitats, while revealing the presence of rare or cryptic species that might otherwise go undetected. These advances continue to reveal new dimensions of animal diversity, while providing insights into the evolutionary processes that have generated the remarkable variety of animal life on Earth. Chapter 5: Ecosystem Diversity and Global Patterns. Ecosystem diversity represents the broadest scale of biological diversity, encompassing the variety of different biological communities and their associated physical environments that characterize different regions of our planet. These ecosystems range from the vast expanses of tropical rainforests and ocean basins to specialized habitats such as caves, hot springs, and alpine meadows, each supporting unique assemblages of organisms while providing essential ecological services that maintain regional and global environmental conditions. Understanding ecosystem diversity requires examining both the factors that determine ecosystem characteristics and the complex interactions among climate, geology, soils, and biotic communities that create the remarkable variety of environments supporting life on Earth. Terrestrial ecosystem diversity reflects the profound influence of climate patterns on vegetation structure and composition, with temperature and precipitation serving as primary determinants of ecosystem characteristics across different latitudes and elevations. Tropical rainforests represent the most diverse terrestrial ecosystems, supporting extraordinary species richness while exhibiting complex vertical stratification that creates numerous microhabitats within forest canopies. These forests occur in regions with consistently warm temperatures and abundant rainfall, enabling year-round plant growth and supporting the intricate food webs that characterize these biogeographically important ecosystems. Temperate forests exhibit seasonal patterns of growth and dormancy that reflect the pronounced climatic seasonality characteristic of mid-latitude regions, with deciduous forests dominating areas with warm summers and cold winters, while evergreen coniferous forests prevail in regions with shorter growing seasons. The seasonal dynamics of temperate forests create temporal diversity in resource availability and habitat conditions that support different species assemblages throughout the year, while generating the spectacular autumn color displays that characterize many temperate regions. Grassland ecosystems develop in regions where precipitation is sufficient to support dense herbaceous vegetation, but insufficient to sustain forest growth, creating vast expanses of grasses and associated herbaceous plants that support large populations of grazing animals. These ecosystems include temperate prairies, tropical savannas, and steppes that have played crucial roles in human history through their conversion to agricultural systems. Grasslands exhibit remarkable resilience to disturbance, including fire and grazing, that help maintain their characteristic vegetation structure while preventing encroachment by woody plants. Desert ecosystems represent extreme adaptations to water limitation, with sparse vegetation adapted for water conservation and efficient utilization of limited precipitation. These ecosystems support surprising diversity despite their harsh conditions, with many species exhibiting remarkable physiological and behavioral adaptations for surviving in environments with extreme temperature fluctuations and limited water availability. Desert organisms often exhibit specialized life cycles that enable them to exploit brief periods of favorable conditions while surviving extended droughts. Arctic and alpine ecosystems face the challenges of short growing seasons, low temperatures, and intense solar radiation that create unique selective pressures favoring small stature, rapid growth, and efficient resource utilization. Tundra vegetation consists primarily of low-growing perennial plants that can photosynthesize and grow rapidly during brief summer seasons while surviving months of darkness and freezing temperatures. These ecosystems support relatively simple food webs, but play crucial roles in global climate regulation through their effects on carbon storage and albedo. Aquatic ecosystem diversity encompasses the variety of freshwater and marine environments that support distinct biological communities adapted to aquatic life. Freshwater ecosystems include rivers, lakes, wetlands, and streams that exhibit enormous diversity in physical and chemical characteristics, while supporting species assemblages adapted to specific hydrological conditions. These ecosystems provide crucial ecological services, including water purification, flood control, and habitat for numerous species, while serving as sources of freshwater for human populations. Marine ecosystem diversity reflects the vast scope of ocean environments that cover over 70% of Earth's surface, while exhibiting remarkable variation in depth, temperature, salinity, and nutrient availability. Coral reefs represent marine biodiversity hotspots that support extraordinary species richness, despite occupying relatively small areas of tropical oceans. These ecosystems depend on symbiotic relationships between corals and photosynthetic algae, while creating complex three-dimensional structures that provide habitat for countless marine species. Open ocean ecosystems support complex food webs based on microscopic phytoplankton that form the foundation for marine productivity, while supporting large populations of fish, marine mammals, and seabirds. These pelagic ecosystems exhibit pronounced vertical zonation, with distinct communities adapted to different depths and light conditions, from the sunlit surface waters where photosynthesis occurs to the dark deep ocean where organisms depend entirely on organic matter produced in surface waters. Deep-sea ecosystems represent some of the most extreme and least understood environments on Earth, supporting unique communities adapted to high pressure, low temperature, and complete darkness. Hydrothermal vents create oases of productivity in the deep ocean through chemosynthetic bacteria that form the base of food webs independent of solar energy. These ecosystems have revealed remarkable organisms, including giant tube worms, unique arthropods, and bacteria capable of surviving under conditions that would quickly kill most other forms of life. Wetland ecosystems represent transition zones between terrestrial and aquatic environments that support unique plant and animal communities, while providing crucial ecological services, including water purification, flood control, and carbon storage. These ecosystems include marshes, swamps, bogs, and other waterlogged environments that exhibit remarkable productivity and support high levels of biodiversity. Wetlands serve as crucial habitat for numerous species, while playing important roles in hydrological cycles and climate regulation. Island ecosystems demonstrate how geographic isolation promotes the evolution of endemic species, while creating unique biological communities that often exhibit high levels of species richness relative to their small areas. Oceanic islands that have never been connected to mainland areas support communities assembled entirely through long-distance dispersal, often resulting in unusual ecological relationships and adaptive radiations. Island biogeography has provided fundamental insights into the factors controlling species richness and community assembly, while highlighting the vulnerability of isolated ecosystems to disturbance. Ecosystem succession describes the predictable changes in community composition that occur following disturbance or during the colonization of new habitats, demonstrating how ecosystems develop and change over time. Primary succession occurs in previously unoccupied areas, such as newly formed volcanic islands or retreating glaciers, while secondary succession occurs in areas where existing communities have been disturbed but soil remains intact. Understanding succession has important implications for ecosystem management and restoration, while revealing the dynamic nature of ecological communities. Ecosystem services encompass the numerous benefits that human societies derive from functioning ecosystems, including provisioning services such as food and freshwater, regulating services such as climate and disease control, cultural services such as recreation and spiritual values, and supporting services such as nutrient cycling and primary productivity. The economic value of ecosystem services has been estimated to exceed global gross domestic product, highlighting the fundamental dependence of human societies on functioning ecosystems while emphasizing the importance of biodiversity conservation. Climate change is altering ecosystem distributions and characteristics worldwide, forcing species migrations and causing shifts in vegetation zones that may lead to novel ecosystem configurations with no historical analogs. Rising temperatures are causing upward shifts in vegetation zones on mountains and poleward migrations in terrestrial and marine species, while changing precipitation patterns are altering the boundaries between different ecosystem types. Ocean acidification and warming are threatening marine ecosystems, particularly coral reefs that are experiencing widespread bleaching and mortality. Human impacts on ecosystem diversity include habitat conversion, fragmentation, pollution, and species introductions that have fundamentally altered most ecosystems worldwide. Agricultural conversion has eliminated vast areas of natural ecosystems, while urbanization continues to fragment remaining natural areas. Understanding these impacts and developing strategies for ecosystem conservation and restoration has become one of the most pressing challenges facing contemporary society as we strive to maintain the ecosystem diversity that supports both biodiversity and human well-being. Protected area networks represent one of the most important strategies for conserving ecosystem diversity, though current protected areas cover less than 15% of terrestrial environments and even smaller proportions of marine environments. Expanding and improving protected area systems while developing sustainable management practices for human-dominated landscapes will be crucial for maintaining ecosystem diversity in the face of growing human populations and increasing environmental pressures. The future of ecosystem diversity will depend on our ability to balance human needs with the requirements of functioning ecosystems that support all life on Earth.